We are Group 5 of bioe120, and our members are Derek Chong, Haley Jones, and Matt Cabernagel, and we were doing a review of osteogenic differentiation of the two distinct subpopulations of human adipose-derived stem cells and an in vitro and in vivo study. The researchers' main goal in this experiment was to compare two populations of human adipose stem cells from fat and choose the one that displayed the highest potential for differentiating or becoming a bone cell. Bone tissue engineering is the regeneration of skeletal tissue. In the past, bone marrow mesenchymal stem cells were commonly used, but now researchers are beginning to use adipose tissue. This is because it is more abundant, easier to obtain, and provides more cells than bone marrow does. Prior experiments by the researchers showed that the two subpopulations chosen would have high osteogenic differentiation potential. They also showed that the scaffolding material would provide for excellent proliferation.
The researchers first prepared the scaffolds or artificial structure for the cells. Then they extracted adipose tissue from women 35 to 45 years old and harvested the adipose stem cells. The subpopulations were then isolated and transfected with green fluorescent protein in order to track them after the in vivo study. The scaffolds were then seeded and implanted into 6 female mice 4 weeks old. After 6 weeks, the tissue was extracted from the mice and analyzed. Some analysis techniques include PCR to assess the gene expression, staining, and micro-CT scans. The results of this will be explained in the results section.
Graph A represents the ASCs seeded in SPCL scaffolds and cultured in vitro with osteogenic medium. Graph B represents ASCs seeded in SPCL scaffolds and implanted in nude mice. Graph C represents ASCs seeded in SPCL scaffolds and cultured in vitro with osteogenic medium. And finally, Graph D represents ASCs seeded in SPCL scaffolds and implanted in nude mice. RUNX2 gene provides instructions for making a protein that is involved in bone and cartilage development and maintenance. The transcription factor osterix is required for osteoblast differentiation and bone formation during development. Osteopontin is a highly phosphorylated cellular protein that is a prominent component of the mineralized extracellular matrices of bone and teeth. Osteocalcin is a similar gene that has to do with the extracellular matrix of bone and teeth. All of these are important in the differentiation structure of bone cells. Figure 2 represents scaffold constructs retrieved from the in vivo experiments and analyzed by micro-CT. These show very clearly that in constructs with ASC STRO1+, the mineralization is much more evident than in constructs seeded with ASC CD29. This attests to the homogeneous distribution of cells into the scaffolds and their active metabolism and osteogenic differentiation in the whole volume of the construct. Figure 3 shows the ASCs producing GFP and relates the presence of cells that were originally seeded to the functionality of the cell scaffold constructs. The presence of the transfected cells within the scaffolds that are implanted in vivo demonstrate that most of the cells did not migrate during the 6 weeks of the in vivo implantation, and that all the results obtained are related to the human ASCs that were originally implanted. When cells were stained, it was apparent that samples implanted exhibited higher fluorescence, indicating a more extensive mineralization shown through Figure 4. As shown through Figure 1, levels differ between in vitro and in vivo. However, ASC CD29 osteopontin was higher in mice, and RUNX2 did not occur in mice. And in ASC STRO1+, they were all overexpressed in mice. Therefore, ASC STRO1+ is more viable than ASC CD29. Transfecting GFP into the cells ensures that after being implanted in the mice, the human adipose stem cells are the cells actually being analyzed and not mouse stem cells. Staining indicated that all the in vivo cells exhibited higher fluorescence compared with the in vitro stem cells. During micro-CT testing, adipose stem cells STRO1+ cells showed more mineralization than adipose stem cells CD29+ cells, and there was calcified tissue. These both indicate a higher osteogenic potential. In the end, the researchers concluded that adipose stem cells STRO1+ cells are more efficient to use in bone tissue engineering due to their high osteogenic.
Only 3 mice were used for each population of human adipose stem cells. This can present problems because it is not an accurate representation of bone tissue engineering in an entire population. It would be beneficial to use more mice so margins of error are decreased. The researchers only chose 2 populations, ASC CD29+ and ASC STRO1+. Comparing these against other populations would be ideal in order to find the population with the absolute best differentiation potential. Limiting the comparison to only 2 populations automatically rules out others without even considering them. When studying the performance of cells, it is always important to give them ample time to display results. If the adipose stem cells were allowed more time in vivo, they may have displayed different characteristics. A nude mouse has a lifespan of 6 to 12 months, so leaving the cells in vivo longer can show the effects of aging on these cells. Nude mice are mice who are bred specifically for laboratory use. They have an altered immune system which allows them to be widely used for applications involving tissue. These mice have no rejection response, allowing them to accept any foreign tissue. The researchers in this experiment not only introduced foreign cells to the mice, but these cells were from humans. The practice of transplanting tissues between species is controversial and may be unethical. The mice used in this study were also sacrificed using cervical dislocation. This technique involves breaking the animal's neck and is considered immoral. Often, these animals can be euthanized or sedated before the procedure, but there is no indication of this in this report.
The researchers used adipose stem cells in their experiment to replicate bone tissue, but they also referenced that adipose stem cells can be used to replicate many other types of tissue, such as cartilage tissue, nerve tissue, muscle cells, liver tissue, and fat tissue. Similar tests like the ones used in this study can be performed to analyze the differentiation potential of the ASCs into these various types of tissues.